Hi, my name's Ed Emmett, and I'm going to talk to you a bit about our latest preprint on bioRxiv, characterization of protease activity during SARS-CoV-2 infection, identifies novel viral cleavage sites, and cellular targets for drug repurposing. So, brief bit of background. My lab is newly established at the Centre for Proteome Research, University of Liverpool, and I'm interested in proteomic approaches and mass spec approaches to study single-stranded positive sense RNA viruses, in particular the coronaviruses and noroviruses, where I've spent most of my career on, as well as other virus families important for human and animal disease, such as the alpha viruses and enteroviruses. I also have interest in mass spec method development, for example, in single cell proteomics. And really within this, the areas I'm most interested in are positive sense RNA virus replication, especially at the single cell proteomic level, cellular targets of viral proteases, the focus of this talk today, and ribosome remodeling during infection. So in this talk, we're using a method called N-terminomics, a proteomic method, to understand proteolysis during SARS-CoV-2 infection. So why, out of all the different aspects of SARS-CoV-2 biology, did we choose proteolysis? So here's a schematic of the SARS-CoV-2 genome and proteome. It's about a 30 kb genome. We have the spike, envelope, membrane, and nucleocapsid structural proteins, accessory proteins, but the bulk of the genome is occupied by these two long polyproteins, PP1A and PP1AB, which are cleaved by the viral proteases NSP3 and NSP5 to make the various proteins that the virus needs for its replication. And this is really a very common method used by diverse single-stranded positive sense RNA viruses. We see it in the alpha viruses, where there's one polyprotein, one protease, and only 3 cleavage sites. Noroviruses, where this gets a bit more complex, we have 6 cleavage sites. To the most complex end, where we can have multiple polyproteins, multiple proteases, and many, many cleavage sites. But in all these cases, these cleavage events and the function of the viral protease is absolutely crucial for efficient virus replication. Takeaway, this is actually already being studied, the role that these proteases and proteolysis play in the replication of viruses, including SARS-CoV-2, is a current field for study. We've seen already structural characterization of the main protease and preliminary work on inhibitors. We've seen the same for the SARS-CoV-2 papain-like protease. And actually, we can extend this. We know it isn't even just viral proteases that are important for efficient coronavirus replication. We actually need cellular proteases as well. For example, cleavage of the spike glycoprotein by cellular proteases TMPRSS2, furin, or cathepsins. So for this reason, we wanted to study a wide variety of cleavage events, proteolytic cleavage events occurring during SARS-CoV-2 infection. If we look at SARS-CoV-2 replication, we can look at RNA levels, protein levels, and titers over a time course. We did this in 2 cell lines, VeroE6, an African green monkey kidney cell line where the virus grows extremely well, though this is perhaps not the most relevant of model systems. It is, however, very widely used for studying virus replication. We also use A549 ACE2 cell line. This is a human lung cell line engineered to overexpress the ACE2 receptor, and it is therefore permissive for SARS-CoV-2 replication. It is perhaps a more relevant model system, however, it does support viral replication at lower levels. We studied infection from 0 to 24 hours post-infection at a multiplicity of 1. If we look at viral protein levels, we do this by mass spectrometry, we see smooth increases from 0 to 24 hours, at least at our time points taken. These were 0, 6, 12, 18, 0, 6, 12, and 24 hours post-infection. RNA levels begin to spike a little later. They spike at 9 hours post-infection, and viral titers we only really see going up at the very end of this time course, at that 24-hour window. So these different time points, 6, 12, and 24, all capture slightly different times and portions of the viral replication cycle. And it was for this reason that we chose these 4 time points for our study of proteolysis. We chose 0 hours at the very beginning, 6, 12, and 24 hours. So how do we actually study proteolysis with proteomic methods? So if we have a theoretical protein X, in this case it has N-terminal acetylation, vast majority of proteins. It also is cleaved by a protease of interest somewhere towards its C terminus. So how can we actually study this with proteomics? Now, in our standard proteomic workflow, what we next do is we incubate all our different proteins with trypsin. This gives us all these additional cleavage sites highlighted here in green, and this means we actually have peptides of appropriate length that we can identify, for example, this peptide here and this peptide here. And these will have potentially non-tryptic cleavage. They won't be preceded by an arginine or lysine, but we have now a large number of other peptides in this sample which we aren't specifically interested in. So protein N-termini include the protein's native N-terminus. This could be acetylated, it could have removal of the N-terminal methionine, or it could, for example, have signal peptide removal. It also include neo-N-termini, which are generated by the activity of proteases within the cell. In this context, it could be viral or cellular. So N-terminomics is the study of all N-termini. Identifying the specific N-terminus or these neo-N-terminal peptides has the advantage that it tells us both what is the protein, so what is protein X, but also the peptide gives us the precise position of this cleavage site. So the very N-terminal residue of that peptide will be the P1 prime residue of any given cleavage site. Trypsin cleavage, as mentioned, generates these new N-termini. So what we need is a method that allows us to selectively identify the N-termini we care about and enrich these over the background that we're generating in the course of our sample preparation through the use of trypsin, or indeed other mass spectrometry compatible proteases. So how do we do this? Well, the key issue is common to most studies of post-translational modifications by mass spectrometry. And really what it is we need a means to allow us to enrich these N-termini. So in our experimental setup, we took our A549ACE2 or Vero E6 cells. These were mock or infected with SARS-CoV-2 for the indicated time points. We lysed these samples and then we blocked all the N-termini in these samples as well as lysine residues by labeling with tandem mass tag reagents. So we used tandem mass tag pro reagents, newly released from Thermo, allowing us to both block these N-termini and quantify the relative abundance of these N-termini in different samples. After we blocked all the N-termini present in the original sample, so all the N-termini we care about, we proceeded to trypsin digestion. So we generate new unblocked N-termini. These unblocked N-termini are then labeled with undecanal. This allows us to deplete undecanal labeled peptides, so those tryptic peptides that we don't want to see, by running these on a C18 column. Everything that doesn't bind, our flow through is therefore enriched for our blocked N-termini, and we can fractionate this by basic reverse phase fractionation prior to injecting our sample onto the mass spectrometer to study these N-terminal peptides. What we found when we performed this, looking at our 24-hour over mock samples, is that we could find significantly regulated cleavage sites or neo-N-termini in SARS-CoV-2 infected cells in both cell lines. Unsurprisingly, those most upregulated corresponded to viral proteins. These proteins shouldn't be present in the mock, so therefore we would expect them to all be highly upregulated. However, we do also see in both cell lines cellular proteins where we identified neo-N-terminal peptides that showed significant enrichment. Firstly, we wanted to study cleavage within viral proteins. So we know that there are cleavage events present in the viral polyprotein and spike glycoprotein. However, we identified far more previously uncharacterized and unknown cleavage sites. Within the nucleocapsid, we identified significant numbers of cleavage events, many of which were common to both cell lines. And indeed, we tended to see a clustering both near the N-terminus and several sites of enrichment, tending to bracket some of the functional domains, for example, the RNA binding or C-terminal dimerization domain. There has been prior published data on cleavage of the SARS-CoV rather than SARS-CoV-2 coronavirus, though not such extensive cleavage. And indeed, there have been further preprints since, for example, from the Robinson group, looking at autolysis of the SARS-CoV-2 nucleocapsid, and indeed incubating that, just leaving this incubating in vitro at room temperature for a week, they find that they start seeing some of the same cleavage sites we see in infected cells. We also see cleavage of the ORF3A viroporin at its N-terminus. A recent cryo-EM study yielded a structure for this viroporin in lipid nanodiscs, so they couldn't resolve the N-terminal amino 39 residues. They generated a deletion mutant, an N-terminal truncation, and this was mislocalized. So this is consistent with signal peptide cleavage and some further processing of this protein. One of the most exciting results we had was cleavage events within the spike glycoprotein. Now, many of these we only spotted in our VaryE6 dataset. This was common to many of our viral proteins. They're much more expressed in the vero cells, so we see more cleavage sites simply because all the viral proteins are more abundant. However, we did see one conserved, entirely novel cleavage site between 636 and 637, located near but N-terminal to the furin cleavage site, as well as a second vero-only cleavage site at 672, 673. Because we had used TMT-based quantification, we were actually able to track the abundance of this cleavage over time. And as you might expect, in line with increases in spike overall, we do see smooth increases in the abundance of this cleaved spike S637 neo N-terminus. We also found cleavage events within other SARS-CoV-2 proteins, for example, signal peptide cleavage of ORF7A, potential cleavage sites in membrane and ORF8, albeit only observed in the vero cells, as well as in novel cleavage sites located within the protein, so internally within NSP1, NSP3, and NSP4. Cleavage within NSP3, for example, would be expected to yield a still active form of the papain-like protease, but it would lack the C-terminal transmembrane domain. So this could be a way to regulate both membrane-based and non-membrane-based cleavage activity of this protease. What could be cleaving these novel cleavage sites within viral proteins? Now, N-terminomics can give us the identity of the proteins being cleaved and the location of the cleavage site. However, mapping a cleavage event to a protease is less straightforward. So what are our candidates? It could be either of the two viral proteases, Mpro or papain-like protease. The reason it likely is not, in the majority of cases, is because these have quite a narrow specificity in terms of the sites they recognize, and these are quite well studied at this point. It could be cellular proteases, and certainly in the case of the spike glycoprotein, we know that cathepsin cleaves spike, but we don't know where. Other candidates include calpains or members of the TMP RSS family. And this is certainly not an exhaustive list, though these are likely candidates. How can we go about testing if any of these proteases are responsible? Well, what we decided to do was to perform viral infection in the presence of specific inhibitors. We took Vero E6 cells, as we saw the vast majority of our cleavage sites in this cell line, and because the virus replicates much better. We allowed infection to proceed uninhibited for 12 hours, because our goal here was not to add inhibitors to halt virus growth. We wanted the virus to replicate, but we wanted to be able to diminish the abundance of specific cleavage events. 12 hours post-infection, we added these inhibitors, high concentration. We allowed infection to proceed a further 12 hours, harvested samples for mass spec, and then processed, as you saw earlier, for N-terminomics. And we used two protease inhibitors. These were camostat mesylate. This inhibits TMP-RSS2 and trypsin, and calpeptin. This is a calpain and cathepsin inhibitor. What we found is under these conditions, the vast majority of viral proteins do not show altered abundance in response to protease inhibitors. We only saw altered abundance of ORF9b and ORF3a, which showed opposite patterns. In the presence of either camostat or calpeptin, we saw reduced abundance of ORF9b, whereas we actually saw increased abundance of ORF3a. And importantly, we didn't see significantly altered abundance of spike or the nucleocapsid protein. However, when we went on to look at the abundance of peptides corresponding to cleavage events within these proteins, we found several, including this N19 in the nucleocapsid and spike 260 and S637, which corresponded to that cleavage site I talked to you about. These showed significant reduction in the abundance of these neo-N-termini in the presence of calpeptin, so this cathepsin and calpain inhibitor. This is consistent with a reduction in the cleavage at that site, which you would expect if these were the proteases responsible. What we actually saw was when we added camostat, we saw an increase in the abundance of these two cleavage sites in spike, though not in the nucleocapsid. And this is really consistent with a model where spike is perhaps preferentially in these cells going through a cathepsin-based entry mechanism, resulting in cleavage at this site here. But if we inhibit cathepsin, we see a reduction in cleavage at those sites. And conversely, if we inhibit alternate routes, for example, TMP-RSS-based routes, we can actually see enhanced cleavage, consistent with the preferential adoption of different entry routes. We wanted to tie down if this cleavage site at 637 had a role in cell entry. Considering our most likely candidate for the protease responsible was cathepsin, where the P2 residue is crucial for cathepsin recognition, we chose to mutate a residue to amino acids upstream of cleavage. So cleavage occurs between 636 and 637, so we chose to make a V635G mutation. What we found was that this mutation showed enhanced entry, albeit a modest phenotype. We actually saw this even in ACE2 cells which overexpressed TMPRSS, albeit the effect was not so significant. What we found when we actually examined these purified lentiviruses that we used for this assay on a gel was that compared to wild type or another mutation, S13A, we saw an increased amount of the cleaved S1 component of spike here. And when we actually performed densitometry on this, this was significantly increased. So it suggests that both the cleavage of this or its ability to be incorporated in lentiviruses is affected by this mutation. And this gives us an entry phenotype. So I've primarily focused on the cleavage of viral proteins during SARS-CoV-2 infection. However, these are not the only cleavage events we see. As mentioned, SARS-CoV-2 encodes two proteases, the main protease Mpro and papain-like protease. We know consensus recognition sequences for both of these. So Mpro cleaves after a APSTV XLQ motif, and the papain-like protease preferentially recognizes a LXGG motif. If we look at the overall abundance of peptides corresponding to these motifs, we find significant enrichment of those corresponding to Mpro motifs. We don't see the same for the papain-like protease. However, there are quite a lot of protease, quite a lot of N-termini with similar sites. So it could be they exist, but are more buried in the noise. If we looked at neo-N-termini, which matched or showed close match to these substrates, we find significant temporal regulation of about 15 proteins. These include kinases such as SARC, WNK, and myosin light chain kinase, as well as a number of other proteins. And these are both cleaved by the main protease, that would be these substrates shown in purple, and by the papain-like protease, so substrates shown in green. And we do see some temporal regulation of these substrates as well. For example, those corresponding to the main protease tend to only show significant upregulation at 24 hours post-infection, whereas those corresponding to the papain-like protease, so SARC here, KLHDC10 and BST1, we're tending to see activity a bit earlier in infection. So we can see significant upregulation at 12 hours post-infection as well, which we aren't really seeing for the Mpro substrates. We can confirm cleavage in an in vitro system of a number of these proteins. So here, these were expressed in a Leishmania-based lysate system. These were incubated with recombinant either PLP or Mpro, and we can see specific cleavage in this in vitro system. And actually, in two of these cases, PINN and SARC, we actually find extra cleavage sites, which were not detected by our initial N-termodynamics approach. We identify cellular proteins being cleaved by viral proteases, but why is this something that is actually useful to know? So there are several reasons why the virus could be cleaving these proteins. It could be an off-target. It might not even matter. It can happen, but it's not very interesting. It could be to inactivate proteins that are inhibitory for the virus, to remove or at least reduce an inhibitory or antiviral response. This would be consistent with the reported cleavage of IRF3, TAB1, and NLRP12, also earlier preprinted by our collaborators. It could result in altered or a gain of function. So really, rather than a sledgehammer, more a fine-tuning of activity, adjusting the activity of cellular proteins that the virus actually needs to better benefit the virus. How can we distinguish between these different possibilities? So we chose to proceed with an sRNA screen. If any of these proteins are inhibitory, then knocking these out should boost viral RNA levels and viral titers. Conversely, if these proteins are actually essential for the virus, but it is simply trying to tune their activity in some way, if we deplete these, it will actually lower viral RNA copies or viral titers. We performed an sRNA screen in A549 ACE2 cells. We added the sRNA. 24 hours later, we added the virus, and we allowed infection to proceed for 72 hours. And what we found is if we looked at viral RNA copies, about 2/3 of our hits showed a significant reduction in viral RNA copies compared to our mock here on the left. And I've divided the hits to those identified originally in the A549 ACE2s, those identified in Vero-E6, and those identified from either cell line, but showing an imperfect match to the consensus sequence. When we looked at viral titers rather than copies, we actually found that every single hit we identified showed a significant reduction compared to the mock. In the majority of cases, this was over 100-fold drop in viral titers produced at the 72-hour time point. We've shown that these proteins are important for the virus. So is this information we can actually use? Well, we know that inhibitors exist against, in particular, kinases, so SARC and myosin light chain kinase. And in the case of SARC, these are inhibitors that are already available and in use in the clinic. We find in particular the inhibitor bifitinib is primarily not a SARC inhibitor, but has well-known off-target effect against SARC. And it inhibits at the sub-micromolar level. We also find we can inhibit SARS-CoV-2 replication using inhibitors targeting myosin light chain kinase. So the ML7 inhibitor works. The MLCK inhibitor peptide 18 doesn't, suggesting that it may be we need to inhibit specific functions of this protein. Several other SARC inhibitors actually gave superior IC50 results, but increased toxicity. And these are all included in the paper in the supplementary data. So to summarize our preprint for you, we used a proteomic method called N-terminomics to study proteolysis during SARS-CoV-2 infection. We found a range of previously unknown cleavage sites in several viral proteins, including the nucleocapsid and spike, which are crucial antigens for vaccination and testing studies. We also found a range of cellular proteins with cleavage sites consistent with cleavage by viral proteases, and they showed temporal regulation across the infection time course, matching the increased abundance of viral proteins. Finally, we showed that if we deplete these cellular proteins, it's inhibitory for the virus, and some of these have commercially available inhibitors in clinical use that can be used to inhibit SARS-CoV-2 infection. If you'd like to read more, this is our preprint, characterization of protease activity during SARS-CoV-2 infection. And you can also see links to our raw data, the code, and the protocols all within the preprint. I'd like to thank my colleagues in the CPR, Claire Ayres, Rob Bainan, Linda Daly, and Philip, all but Rob, who were actually contributed to this paper. Rob did give very good feedback as well. The Vignusi lab, especially first author on this paper, Bjorn Meyer, who, without which I couldn't have done this work. He did all the infection work for this paper. The Chakrabarti lab, also at the Pasteur, including PhD student Stacy, who did the pseudotype work characterizing cell entry of that V635G mutant. Paters, who helped us looking at Src kinase. The Go lab, who performed sRNA and inhibitor screens. Charlie Craig and the Verdin lab, who helped us validate in a cell-based assay some of our cleavage events. And Jan Gambin and Emma Sierecki at University of New South Wales, who provided the Src cleavage and the in vitro data that I've shown you. Thank you for listening. I'll happily take questions at my email here, or you can find me on Twitter @edemmett. Thanks a lot.